Hi guys, we are going to start with our chapter number 5, Enzymes. It says the key learning objectives. It include understand enzymes functions as a biological catalyst, investigate how temperature and pH impact enzyme activity, and then the third, explore enzyme specificity. Let's start with fundamental enzyme concepts. What are enzymes? Definition: protein molecules that act as biological catalyst, okay, called as enzymes. Critical role of enzymes are: it accelerates metabolic reaction which are essential for sustaining life. Some unique characteristics include highly specific molecular structure, contains a specialized active cell, enables chemical reaction to occur rapidly. Biology is an understanding subject, but you have to learn also several concepts here. Alright. Next is enzyme structure and function. Alright, active site of enzymes is specific three-dimensional shape, complementary to substrate molecule, determines enzymes unique catalytic properties. Enzyme substrate interaction. Let's talk about temperature and enzyme activity. Kinetic energy relationship. Low temperature at 0 degrees centigrade, minimal molecular movement you will see, rare substrate enzyme collision, and extremely low reaction rates. Why? Because temperature is 0 degrees centigrade. Next, increasing temperature, enhanced molecular kinetic energy, more frequent substrate enzyme interaction. So we need higher temperature for the enzymes to be reactive. It also increased reaction efficiency. When the temperature is increased, then reaction efficiency also enhances. Optimum temperature. Human body enzymes at 37 degrees centigrade. Plant enzymes lower temperatures. Bacterial enzymes can tolerate up to 80 degrees centigrade. Denaturation process, temperature threshold is at 60 degrees centigrade. Mechanism, excessive heat disrupts enzyme molecular structure. Active site loses complementary shape and enzymes become non-functional. So our threshold of temperature is 60 degree centigrade, not more. Alright. Next we have pH and enzyme functionality. pH sensitivity, optimum pH is typically around 7. Variations. Some enzymes function best in acidic conditions. Stomach enzymes can operate at pH of 2. Denaturation mechanism, pH significantly outside optimum range, yani more than 60, causes molecular shape alteration, also prevent substrate binding capabilities. Then we have lactose intolerance. Enzyme example, lactase enzyme function. It breaks down lactose into milk. Production abundant in infants, decreases in most adult, genetic variation in certain population. Consequences of lactase deficiency. Undigested lactose remains in digestive system. Trigger osmotic water movements. Potential symptoms are, for this deficiency, lactase deficiency, diarrhea, abdominal discomfort, and flatulence. Alright, now let's talk about experimental consideration investigating enzyme activity. Measurement techniques we have includes temperature variation, pH manipulation, and reaction time tracking. Precision in recording, use whole degree, 0.5 degree temperature readings, record times to nearest second, minimize experimental errors. So now we will study enzymes characteristics, description and impact. First characteristic is active site, description is complementary substrate shape, impact determines specificity. Next characteristic is optimum temperature. It differs species and is specific according to the species. Maximize reaction rate. Third is optimum pH. It typically near neutral, maintains structural integrity. Then denaturation, structure breakdown, and stops enzymatic function. So key takeaways for us are: enzymes are critical for biological processes. Structural integrity determines functionality. Temperature and pH significantly influence enzyme performance. Next we have biological catalyst and their functionality. Introduction to enzymes, again definition: biological catalysts that speed up metabolic reactions, critical for sustaining life by enabling chemical reactions that would otherwise occur very slowly or not at all. So enzymes actually enable them to do the chemical reactions. Found in all living organisms including plants and animals. What are the characteristics of enzymes? Naming convention. Name typically ends in ase. Named based on the reaction they catalyze. Carbohydrases break down carbohydrates. Proteases break down proteins. Lipases break down fat and oils. Specificity and mechanism. Enzyme specificity. Each enzyme works with only one type of substrate, remember it. Active site has a unique shape that perfectly matches the substrate. Substrate must have a complementary shape to fit into the active site. Key components: substrate, the molecule the enzyme acts upon; active site, specific region where substrate binds; enzyme substrate complex, temporary structure formed during reactions; and then products, new molecule created after enzyme catalysis. Enzyme activity factors. Number 1, temperature effects. It impacts enzyme activity through kinetic energy, frequency of effective collisions, and potential for denaturation at extreme temperatures. Second is pH effects. Different enzymes have optimal pH ranges. Extreme pH can cause altered enzyme shape and potential denaturation. Industrial and biological applications used in various industries, critical for metabolic processes. Some examples are amylase breaking down starch, catalase decomposing hydrogen peroxide, and the synthesis of protein. Performance table of enzymes characteristics. Enzyme amylase, its substrate is starch. Function is to break down carbohydrate, and the optimal condition should be neutral pH, moderate temperature. Second is protease. Substrate is protein. Function is breakdown of protein. Optimal condition, it varies by specific enzymes. Third is catalase. Substrate is hydrogen peroxide. Function is decomposition, and then optimal condition should be cellular environment. So our key takeaways are: enzymes are essential for life, highly specific and efficient, sensitive to environmental conditions, and crucial in metabolic processes. Experimental consideration: when studying enzymes, control variables are important. You have control variables like temperature, pH, substrate concentration, enzyme concentration, and volume of solutions. Some fact about the enzyme is one catalase molecule can break down 4 million hydrogen peroxide molecules in just 1 second. I think it's 44 million. Okay. Alright, next is experimental skills in enzyme activity. Investigating pH effects and catalase activity. Experiment overview. Purpose: determine how pH impact catalase enzyme activity. For this we can use the key enzyme catalase. It breaks down hydrogen peroxide into water and oxygen. The technique is measure time for filter paper to rise in hydrogen peroxide solution. Materials needed: fresh plant material, celery or potato, blender or pestle and mortar, distilled water, 3% hydrogen peroxide solution, buffer solution at different pH levels, filter paper squares, forceps, stopwatch, at least 3 to 4. Remember it, it comes in the short question. Maybe they will ask you to write some of the materials, okay, to test this catalase activity. Experimental method. Number 1, catalase solution preparation. Blend plant material with water, strain through muslin or sieve, collect liquid catalase extract. Next, procedural steps: label beakers with different pH levels, add hydrogen peroxide and buffer solution, dip filter paper in catalase solution, drop paper into hydrogen peroxide. Then time: see how long paper takes to rise. Key observation: oxygen bubble will cause filter paper to float. Different pH levels affect enzymes reaction speed. This will be the key outputs. Next, investigating temperature effects on lipase activity. Alright. Now enzyme overview. Which one? Lipase. So we are talking about enzyme breaking down lipids. Reaction product: fatty acids and glycerol. Materials that we need: fresh milk, lipase solution, sodium carbonate solution, thymol phthalate indicator, test tubes, water baths at different temperatures. Experimental techniques: use color change indicator, thymol phthalate, blue in alkaline condition, loses color as pH become less alkaline. Procedure: prepare test tubes with milk, sodium carbonate indicator, place in water baths at different temperature, add lipase solution. Time: noted down the color change and disappearance, and then you can also make the graph to see, like to exactly catch what are the disappearances in the time intervals. Key observation: initial blue color indicates alkaline condition. Color loss shows fatty acid production. Temperature impacts reaction speed. You can make a graph. On x-axis you can keep the temperature, on y you can put time for color change. Some safety consideration which you should all also learn, maybe 3 marks or more. Careful handling of hot water baths. Use test tubes holders. Maintain laboratory safety protocols. Experimental evaluation. Analyze factors affecting enzyme activity. Identify potential sources of experimental error. Suggest improvements for precision. Alright, with this we have finished our this chapter, and now we are going to continue with our chapter number 6. Thanks. See you in our next class.